Hello, my name is Monica McDaniel, and today I am going to talk about the effects of calcium, chloride, and fluoride on oxygen evolution rates and tyrosine Z radical signal. Photosystem II is a part of photosynthesis that produces oxygen. For this project, I worked with Dr. Haddy at the University of North Carolina at Greensboro. This presentation is going to be an overview of this poster. The purpose of this experiment was to determine the effects of calcium, chloride, and fluoride on oxygen evolution rates and the tyrosine Z signal. Knowing that fluoride is a known inhibitor that increases the tyrosine Z signal, we compared a sample with fluoride to samples with and without calcium. We observed the dependence of calcium on oxygen evolution by comparing a sample that contained calcium to a sample that did not contain calcium. It shows the electron transfer site. Tyrosine Z is in line for electron transfer, while tyrosine D is not. In this experiment, the Photosystem II samples were prepared from fresh market spinach by extraction with Tytrin X100. The samples were prepared with different buffers to compare the effects on the tyrosine Z radical signal and on oxygen evolution. The picture shows an EPR spectrometer. EPR is a technique which depends on an unpaired electron, and you observe transitions in a magnetic field. Tyrosine Z is induced by illumination and decays during dark adaptation, leaving only tyrosine D. Fluoride is a known inhibitor that increases the tyrosine Z signal. Therefore, it was used as the control. Taking a look at the graphs, the top graph shows spectra for a sample with 100 millimolar concentration of fluoride, and it compares it when illuminated and when dark adapted. Since we know that only the tyrosine D radical is present after dark adaptation, we know that the increase in signal is tyrosine Z. By comparing the peak heights, we were able to measure the increase in tyrosine Z signal. In the second graph, it shows the increase in tyrosine Z signal after illumination. The 100 millimolar fluoride sample had a 51% increase in signal, and the sample containing no calcium had a 55% increase. Therefore, it is evident that the absence of calcium resulted in inhibition of electron transfer. For the oxygen evolution part of the experiment, one sample contained calcium chloride and the other contained sodium chloride. The graph shows the oxygen evolution rates of the samples with and without subunits that restrict the exchange of calcium ions. The samples containing calcium had higher oxygen evolution rates compared to without calcium. It is shown that calcium is required for activity. In conclusion, we found that the tyrosine Z signal at 77 degrees Kelvin increased in the absence of calcium, which shows that the electron transfer was inhibited. It was known that the signal would increase with fluoride because it is a known inhibitor. Photosystem II, in the presence of calcium chloride, did not produce a tyrosine Z radical signal. Therefore, it can be determined that calcium is required for electron transfer as well as oxygen evolution. These were the references used during this experiment. Thank you for taking the time to listen to this presentation.